students, in continuation of my last class, today I will be talking about viruses. As I have told you earlier that viruses are the controversial creature in this universe, and as long as they are outside of the living cell, they are considered as non-living, and when they come in contact with the host cell, they do certain activities for which the scientists are compelled to believe that virus is a living particle. So, viruses are a very small tiny particles. They are invade and reproduce only when they come in contact with the living cell. They do not use any energy to grow or respond to the surroundings. They do not use or make food or produce any waste, which makes us to think that virus is non-living. Now, if we see some novel properties of the viruses, we will find that they are obligate intracellular parasite, and when they come in contact with any living cell like bacteria, protozoa, fungi, algae, plant and animal, they do their certain activities, and we consider that time these viruses are living. They are ultra microscopic in size. Their size ranges from 20 nanometer to 450 nanometer. Now, when in my last class I was talking to you about the prokaryotic organism, and bacteria was one of such example, I told you that the sizes of bacteria vary from in the tune of micrometer level. But here the viruses are so tiny that their structure, their sizes vary in the tune of nanometer. They are very simple in structure. They do not independently fulfill the characteristics of life. They are inactive macromolecules outside of the host cell and only active when it comes in contact with the host cell. They can form crystal-like masses, and basic structure consists of a protein cell which is called the capsid, which is surrounded by the nucleic acid core. So, what we can say that these viruses are a very tiny particle and they are very simple in structure. This nucleic acid are either DNA or RNA molecules. Nucleic acid can be the double-stranded DNA, single-stranded DNA, single-stranded RNA or double-stranded RNA. But both RNA and DNA are not present in any virus particle. The molecules on virus surface impart high specificity for attachment to the host cell. They are multiple by taking control of the host cell's genetic material and regulate the synthesis and assembly of the new virus particle inside the host. In reality, they lack the machineries for synthesizing the protein molecule, and they also lack the enzymes for most of the metabolic processes. So, if we see the sizes of viruses, then we can find that the biggest virus till date reported is the pox virus, and the smallest one is the yellow fever virus. The size of the pox virus is in the range of 250 nanometer, whereas yellow fever virus is 22 nanometer. Now, as I have told that they are very tiny small organism, and if we see its architecture, then we can find that this viruses has two major part. One is the covering part, another is the central core. This covering part can be once again divided into two. One is called the capsid and another is the envelope, and this envelope is also not present in every viruses. It is found in some of the viruses. If we see the central core, we can find that this is the nucleic acid molecule, either DNA or RNA, which is covered by this capsid. And these virus protein are also present along with this nucleic acid. And now let us see the architecture of viral cell. Now, if we see this virus particle, we can find that inside the cell these nucleic acids are present, which is covered by the protein coat, which is called the capsid or protein coat. Sometime in some viruses, some lipid envelope are present, which are bilayer in nature, and sometimes some glycoprotein receptors are present on the cell surface of the viruses, which help the virus to recognize the host cell. These are the different architecture of bacteria, virus, sorry, and this virus architecture, if we see in some of the viruses, this protein coat is there. Beside this protein coat and nucleic acid, there are certain envelopes are present to which some spike-like structures are present, and this spike-like structure, it helps the virus to go and sit on the cell surface of the bacteria or the host cell. Now, if we see the shape of virus particle, then based on the shape, viruses can be categorized into different grades. One is called the helical type of helical shaped virus. Now, in this helical shape, the structure itself indicates that it is helix like structure. Here this nucleic acid is present as the genetic material, which is covered by the protein coat, which is called the capsid, and altogether this is called the nucleocapsid. This nucleocapsid is present inside the virus, and it is covered by a layer which is called the envelope, and this particle, this as its shape is helical like, this is called helical shaped viruses. Another group of virus is called icosahedral. Now, here this type of viruses under low magnification looks like spherical, but when under proper magnification it looks like icosahedral. Here also inside this nucleic acid moieties are there, which is covered by the capsid or the protein coat, and some matrix molecule is there inside, and sometimes it is enveloped, and this envelope has also in this envelope some spikes are present. So, this is the icosahedral enveloped virus. Another group of virus is called the complex virus. Now, here pox virus and bacteriophage are the example of such type of viruses. In case of complex virus, this nucleic acid moieties are present, along with that the outer envelope is present, in which the soluble protein and antigens are present, lateral body is also there, and the size of this virus is 2 50 nanometer to 300 nanometer, which is considered to be the biggest virus. Now, if we come to another complex virus, then we can divide the entire structure of such type of viruses into two major groups. One is called the head, another is the tail. Now, in this head portion, this head is covered with a capsid, that means protein coat in which the nucleic acid is present. Now, this head is attached to the tail with the collar and sheath, and in this tail portion there is a base plate to which some tail fibers are present along with the tail pins. These tail pins, they help, and these fibers, they help the virus to go and sit on the cell wall of any bacteria or host cell. Now, this is the architecture of a virus, and if we see the life pattern or the nature of replication of such viruses, the entire activities can be divided into 6 major groups. One is called the adsorption, where this is the phenomena where binding of virus to the specific host cell surface is taking place, followed by penetration, then replication, assembly, maturation, followed by release. Now, how these activities are going on inside any host cell, let us take some example and let us study that. Now, bacteriophage, as I have told you, that it is a complex virus and it has got one head, another tail, and tail has got some tail pins which help the virus particle to sit on the wall of the bacteria. Now, when we are just considering E. coli as the bacteria, and this E. coli or the bacteria is getting infected, getting killed by this virus infection. Now, when the first virus particle is coming and sitting on the cell wall of this bacteria, then first is the adsorption. So, it is coming and getting adsorbed on the cell wall of the bacteria, outside wall of the bacteria. As soon as it sits on the wall of this bacteria, the next step is that it inject the genetic material, that is the nucleic acid which is present in the head portion, which is covered by the capsid, and they inject that nucleic acid material inside the bacterial cell. So, this nucleic acid is inserted in the bacterial cell. The other portion of this virus particle is staying outside. Now, as soon as this nucleic acid enters to this bacterial cell, it is called the penetration. Now, once it penetrated to the bacterial cell, it starts its replication. Now, this virus particle is inserted in the bacterial cell, and by mistake this bacterial genetic material start recognizing this virus nucleic acid as part of them, and then the replication starts and they start synthesizing the different body parts of the virus molecule, and here they synthesize the different part. It is the replication followed by the assembly. In the next step, they start assembling the different part of their body and they take the complete shape of a virus particle, and this is called the maturation phase. Now, after assembly, the maturation takes place when a million and millions of these virus particles are matured inside the cell. They put pressure on the cell surface from inside and ultimately it got burst and all the virus particles are going out of the cell, resulting the killing of the bacterial cell, and this is called the lytic cycle of virus. Now, this is one type of life cycle what viruses they do, they perform. Another type of life cycle the virus particle which is observed is the lysogenic pathway. In this lysogenic pathway, unlike these two steps up to penetration, this virus genetic material enters to the bacterial cell, and then this bacterial genetic material which is present, they start recognizing the virus nucleic acid as part of their genetic material, and it gets assembled within the bacterial genetic material. Now, here when they undergo the normal cell cycle, cell division or reproduction, this virus nucleic acid is also getting divided into the daughter cell, and in this way the life cycle goes on in the bacteria. Now, in this way few generations can be taken place, and it is remaining as part of the genetic material of the bacterial cell. But under any certain environment, suppose exposure of UV light or any drastic condition, once this particular genetic material starts getting expressed, then the virus nucleic acid, they once again starts their construction. That means they start synthesizing their, this replication process starts, and they synthesize the different part of the virus particle, and then they get assembled, and once they mature they release, resulting the death of the bacterial cell. And this type of life cycle is called the lysogenic pathway, and these are the two major life cycles which are followed by the virus particle. Now, if we see, if we classify these viruses, we can divide this virus particle into two major groups. One is called the plant virus, another is called the animal virus. The tobacco mosaic virus is one of such example of plant viruses, and animal viruses are the pox virus, herpes, yellow fever and so on, HIV and so on. Those viruses are the animal virus. Now, whatever may be the virus particle, these viruses are the smallest creature which are mostly pathogenic in nature. Now, if we classify these viruses based on its genome structure, then I have already mentioned that DNA, either this is the DNA based viruses or RNA based viruses. Now, when it is DNA based viruses, either it is the double stranded DNA or the single stranded DNA, it is double stranded reverse transcriptase DNA or double stranded RNA, that reovirus is one of such example, polar single stranded RNA, that is Toga virus, negatively polar single stranded RNA, that is the rhabdovirus, and single stranded RNA reverse transcriptase is the retrovirus. So, these are all about these viruses. So, these viruses are the tiny and the smallest creature in this universe, and with this I am just concluding my this prokaryotic and this organism, and now let me switch over to the eukaryotic organism and eukaryotic cell structure and its cell organelles. Now, here in this particular cell organelles, I will be mainly concentrating to the cell theory. Now, as I have already talked to you about the microbial cell, prokaryotic, eukaryotic cell, I have also mentioned you the discovery of microscope and existence of this micro creature in this universe. Now, first in 1665, the scientist Robert Hooke first visualized the dead cork cell under the microscope, and from there the studies or the existence of the cell in this living creature came into picture. In the year 1838 and 1839, two different scientists, Matthias Schleiden and Theodor Schwann, they proposed the cell theory. Now, what the cell theory states? The cell theory states that all organisms are made up of one or more cells and the products of those cells. All cells carry out live activities. It requires energy, grow and have a limited size. New cells arise only after other living cells by the process of cell division. That means, cell division is one of the living activities of the cell. And obviously, the question comes that in our body, in your body, how many cells are present? Obviously, we are eukaryotic and multicellular organism. So, obviously, how many cells are there in our system, in our body? It is 50 million. That means, it is in the tune of trillions of cells. And not only that, they are of different types. The cells are diverse in structure as well as in function. But if we see the basic type of cell, then we can find that the entire cell can be divided into two broad categories. One is the prokaryotic or primitive nucleus cell, or eukaryotic or the true nucleus cell. Now, I have already talked to you about the prokaryotic cells in details. Now, let me tell about the eukaryotic cell in this particular class. Now, as I have already mentioned that eukaryotic cells are the developed cell. Obviously, when we are talking about the development, at the same time we are talking about the complexity. Now, when we are talking about this complexity, and if we see, the prokaryotic cells are the predecessor of the eukaryotic cell. Eukaryotic cells, as in my first lecture, I have told the 5 kingdom classification, and there we started the eukaryotes from protists, then fungi, plant and animal. So, these all belongs to this eukaryotic cell. Today, I will try to cover the cell and its structure and different organelles present in this eukaryotic system. Eukaryotic organism can either be single cell or multi-cell depending upon its development. These eukaryotic cells have a membrane bound nucleus, and different organelles which are present in this eukaryotic system is the nucleus, mitochondria, chloroplast, smooth and rough endoplasmic reticulum, Golgi apparatus, ribosome, lysosomes, vacuole, paroxysm and so on. So, let us start the different cell organelles. Now, when we are talking about the different cell organelles, let me start with the protoplasm. This is the fluid which is present in all living systems. Now, protoplasm is the living content of the cell that is surrounded by a plasma membrane. Now, protoplasm in living system is composed of cytoplasm and nucleoplasm. Now, when we are talking about the protoplasm, it is the liquid or the fluid, and you know, in each living cell more than 70% cell contains this type of fluid. That means 3/4 part of the cell contains the fluid. It is essential for any living system. Now, when we are talking about this protoplasm is the fluid where all the cell organelles are present. When we are talking about the only fluid minus the organelles are the cytoplasm. And when we are talking about the fluid which is present inside the nucleus is called the nucleoplasm. And when we are talking about the cell sap, including all the organelles present inside the cell is the protoplasm. Protoplasm is also composed of a mixture of small molecules such as ions, amino acids, monosaccharide and water, and the macromolecules such as nucleic acid, protein, lipid and polysaccharide. They are the macromolecules, this nucleic acid, protein, lipid and polysaccharides. I will be talking to you in details about each of these macromolecules and their function, but these particular macromolecules are also present along with some micromolecules in the protoplasmic fluid. Now, let me come to the cytoplasm. As I have already told that the portion of cell enclosed by the cell membrane, but not any organelles of the cell is the cytoplasmic fluid or the cytosol. It can be divided into compartment through complicated array of internal membrane. This cytoplasm, in this cytoplasm there are different organelles are present. Cytoskeleton, ribosome, metabolic enzymes etc. are present in this cytoplasmic fluid. Next, let me come to the most important organelle in the cell. This is the nucleus, which is considered to be the brain of the cell. It is the largest organelle of the cell. Robert Brown in the year of 1831 first discovered this cell. The cell contains the hereditary apparatus which is called the chromosome. It has got the envelope which is bilayer, that means two bilayer membranes are there. Now, if we see the function of two bilayer membrane, then we can find that double membrane bag of the nucleus has got outer membrane and inner membrane, and it contains the nuclear pore which is not there in prokaryotic organism. In prokaryotes there is no cell membrane, that nuclear membrane which is covering in the genetic material which is present. Nuclear pores in case of eukaryotic cell are placed for selective transport between the cytoplasm and inside the nucleus, that is protein and messenger RNA. The outer membrane is continuous with the rough endoplasmic reticulum and has ribosome attached to this. Now, if this is the nucleus, nucleus has got one membrane and it is the outer membrane. The outer membrane is attached to the endoplasmic reticulum, and here this is the two layer of your this nuclear membrane. The space between the outer and the inner membrane is continuous with the rough endoplasmic reticulum. So, if some ribosomes are attached to it are called the rough endoplasmic reticulum. So, it is present in the nucleus. The function of the nuclear envelope is that the nuclear envelope separates the nucleoplasm from the cytoplasm. Now, here the outside of this liquid is the cytoplasm, and here this barrier, it separates the inside liquid from the outside cytoplasmic liquid. So, inside whatever liquid is there is the nucleoplasm, and here whatever liquid is there, it is the cytoplasm. And if this is the total cell structure, it separates DNA replication and transcription in the nucleus from RNA translation in the cytoplasmic fluid. Here RNA translation is taken place, and here DNA replication is taken place. So, this is the main difference in the activities of the nucleus. Now, this nucleus has got some nuclear pore. This nuclear pore is formed at the site where inner and outer membrane are pinched together. So, this inner and outer membrane are pinched together is the nuclear pore. So, it is 100 nanometer in diameter. This is 100 nanometer in diameter. It is filled with the filamentous material. The nuclear pore is a selective filter for the transport of larger molecule. A ring of 8 subunits are present in this particular nuclear pore, and each subunit projects like a spoke-like unit into the center, so that the pore looks like a wheel with 8 spokes from the top. So, from the top view, it looks like a wheel of a cycle, and inside the central plug is there, which is made up of a protein. So, this is the function, this is the structure of nuclear pore. So, obviously, what is the function of nuclear pore? This nuclear pore is mainly import the protein and export the RNA for RNA translation. If we see the structure of nucleolus, we will find that the nucleolus is a diverse, densely stained structure found in the nucleus. It is not surrounded by the membrane, and it is highly visible region in the nucleus consisting of the aggregation of ribosomal RNA and ribosomal protein. The function of nucleus, it plays a very important role. Nucleus controls the transfer and replication of hereditary molecules, that is DNA and RNA, between the parent cell and the child cell. That means, during cell division, the distribution of genetic material from the parent cell to the daughter cell is controlled by the nucleus. Nucleus ensure equal distribution of exact copying of the genetic content during the process of cell replication, and this is the main function of nucleus in the animal system. Nucleus is the place for DNA transcription, for the production of messenger RNA, which synthesize the protein. The process of energy and nutrient metabolism in the cell is regulated by the nucleus. Nucleus is responsible for the secretion of ribosome. Now, immediately which is connected organelle to the cell, this nucleus is the endoplasmic reticulum. Endoplasmic reticulum is a network of interconnected closed membrane vesicle. It is a single membrane organelle. It divides the cell into compartment, and it is the site for protein and phospholipid synthesis. It is connected with the nuclear envelope. Now, this endoplasmic reticulum can be of two types. One is called rough endoplasmic reticulum, another is the smooth endoplasmic reticulum. Rough endoplasmic reticulum, so where ribosome is attached to the surface of the endoplasmic reticulum for the synthesis of protein. It may not be permanently attached to the endoplasmic reticulum. Attached to this ribosome is synthesized the membrane and the secretory protein. Secretory protein pass through the rough endoplasmic reticulum membrane and enters the rough endoplasmic reticulum cavity, that is called the lumen. And it is the site for glycoprotein synthesis, as the enzyme requires the presence of endoplasmic reticulum. The function of rough endoplasmic reticulum is that it is the main organelle for the production and processing of specific protein at ribosomal site that are later exported. The protein folding is an also important event of rough endoplasmic reticulum. Rough endoplasmic reticulum is also transport this protein to the site where they require. As and when it is required, they transport those protein to that particular site. When we are talking about the smooth endoplasmic reticulum, this is the site for fatty acid and phospholipid synthesis. Calcium storage is also another activity of the smooth endoplasmic reticulum. It lacks ribosome, it is tubular in appearance, and smooth endoplasmic reticulum contain enzyme that synthesize lipids including membrane phospholipids, cholesterol and hormones. The function of smooth endoplasmic reticulum is that it produces enzyme for the synthesis of lipid, phospholipid, steroids and hormones. It is the site for metabolism of carbohydrate. It detoxifies the drugs and proteins and contraction of muscle cell. Now, coming to the next organelle is the ribosome. Ribosome are the component of cell that makes the protein from the amino acids. The DNA sequences in genus is copied into the messenger RNA, that is the mRNA, and ribosome then read those information in this RNA and use it for the production of protein. That means 3 coded nucleotide is read by this messenger RNA and they synthesize this protein. Ribosome are made from the complex RNA and protein, and it has got 2 major subunit. If we see the composition, 65% it is composed of 65% of ribosomal RNA and 35% of ribosomal protein, and its size varies from 20, the ribosome is of 20 nanometer in diameter. Eukaryotes have 80S ribosome, where the small unit is the 40S and larger unit is the 60S. It is different from that of the prokaryotic system, where we have the 70S ribosome, the smaller unit is the 30S and larger unit is the 50S in case of prokaryotic. But here in case of eukaryote, the smaller one is 40S and larger one is 60S subunit. Now, if we see the function of ribosome, we can find that ribosome are the workhorses of protein biosynthesis. It uses the messenger RNA as a template, and the ribosome transfers each codon, that 3 nucleotide of the mRNA, pairing it with the appropriate amino acid provided by the transfer RNA, and its main function is the protein synthesis. Now, coming to the next important organelles which is present in the cell is the Golgi apparatus. It is the stack of flattened membrane bound sacs that look like a stack of pita breads. Some cells have one large stack, other may have hundreds of small stacks. It is the transport vesicle carry protein from the endoplasmic reticulum to the Golgi apparatus. So, nucleus to endoplasmic reticulum to Golgi apparatus. Now, the number of Golgi bodies in the cell in a cell depends upon the level of its activity, that means how efficiently a cell is performing or doing its activity. The more activity, the greater is the number of Golgi bodies. So, more active the cell, number of Golgi complexes are more. If we see the function of Golgi bodies, then we can find that it is the interaction with other organelles, but is not connected by a membrane with the other organelles. Chemically modify the secretory proteins and lipid transported from endoplasmic reticulum in the transport vesicle. This Golgi bodies has got cis and trans phase and modify the protein by the cell, which is package and export the protein. The interfunction of this organelle is that it transfers the vesicle to carry the secretory protein from endoplasmic reticulum to the Golgi apparatus. The rough endoplasmic reticulum and Golgi apparatus also maintain the cell membrane, the structure of cell membrane. That means, what is the major function of these Golgi bodies? The protein which is synthesized by the endoplasmic reticulum is coming and getting transported to the Golgi apparatus, and this particular organelle help in transportation of this protein and lipid. The most important organelle, one of the most important organelle of the cell is the mitochondria, which is otherwise known as the powerhouse of the cell. It is the place of oxidative metabolism and ATP production. The tubular sausage shaped organelle enveloped by a double membrane. It contains its own DNA and ribosome, which is called mitochondrial DNA and mitochondrial ribosome. This mitochondria is bilayer organelle, which has got an outer membrane and inner membrane, and in between the space of outer and inner membrane is the intermembrane space. Mitochondria is the site of all enzymes for Krebs cycle, and it contains, as I have mentioned, that mitochondrial DNA and ribosome. This is the structure of mitochondria. It has got the inner membrane and the outer membrane, and this is the site for ATP synthesis, and this is the intermembrane space, and this is the ribosome which is present, and these are the granule, and this is the most important, that is the secrecy of the mitochondria. If we see the function of mitochondria, then it is the organelle where energy conversion is taking place. This is the site for citric acid cycle or Krebs cycle. It is the site for electron transport chain and oxidative phosphorylation. That means, mainly the respiratory process is taking place in this organelle. Here heat production is also taking place, and it is also the storage of calcium ion. Now, coming to the next important organelle is the chloroplast. The word chloroplast is derived from the Greek word where chloros means green and plast means the form. It is otherwise known as the plastid. Now, in many cells, it is mainly found in the plant cell or some of the microorganisms which are photosynthetic in nature. This is the organelle which helps in photosynthesis. The chloroplast converts the light energy to chemical energy in the form of ATP and NADP to NADPH. Now, a typical cell organs which contains about 10 to 100 of chloroplast molecule. The chloroplast, if we see the size, it is a flat disc like structure and its diameter varies from 2 to 10 micrometer and thickness is 1 micrometer. It contains one or more molecules of small circular DNA and ribosome within it. If we see the structure of this chloroplast, we can find that this is here the thylakoid membranes are present in the chloroplast, and thylakoid is a membrane bound compartment inside the chloroplast which contains light absorbing pigment which is the chlorophyll and carotenoid. See the structure of chlorophyll, it is a very complex structure. It stacks the thylakoid. The stacks of thylakoid is called the grana, and the matrix of chloroplast within the grana are embedded is called stroma. Photosynthesis takes place on the thylakoid membrane, whereas mitochondria is the site for oxidative phosphorylation. So, this is two major difference between the chloroplast and the mitochondria. Now, next important organelle which is present in the cell is the lysosome. Lysosome is also otherwise known as the digestive organelle, which is single membrane bound organelles. If we see the nature, it is the acidic organelle and its pH varies from pH is in the range of 5 in the membrane. It contains the degradative enzyme such as the acid hydrolase. The major function of lysosome is that it is the digestive plant of the plant lipid and carbohydrate, and it helps in transportation of undigested material to the cell membrane for its removal. It varies in shape and depending on the process being carried out. Now, this particular organelle is otherwise known as the suicidal bag. If by any chance the membrane of this lysosome is getting cracked, its all digestive enzymes come in contact with the other organelles and it kills the total cell. Now, let me come to the another important organelle, which is the paroxysome. This is responsible for the oxidation of fatty acid. No ATP production is taking place in this particular organelle, but just the heat production is there. It protects the cell from hydrogen peroxide, that means from oxidation, as it contains the peroxidase enzyme. Besides this oxidoreductase group of enzyme, it contains oxidases and catalase group of enzymes too. It is a single membrane organelles. If we see the function of paroxysm, its major function is to break down the fatty acid molecule through beta oxidation. Paroxysome stores the enzyme that protect the cell from toxic peroxides. Now, the another organelle which I will be talking is the cytoskeleton. Now, cytoskeleton is the interior framework of the cell. Now, when we are talking about the cell and when it undergoes different activities, the network of protein filaments which lend support to the cell as well as its effect on the cell movement is the cytoskeleton material. The dynamic structure, it has got a dynamic structure and it maintains the shell shape, enables the cellular motion and plays some important role in intracellular transport and the cell division. Cytoskeleton consists of threse major kind of filament, which are microfilament, intermediate filaments and microtubules. These are mainly composed of the protein polymers. Now, I have talked to you about the different organelles which are present in the eukaryotic cell. Besides this organelle, the cell may contain autophagosome. The main function is the vesicle which sequesters the cytoplasmic material and organelles for the degradation. It is a doubled membrane compartment and it is present in all eukaryotic cell. The another organelle which is present is the centriole. It is the anchor of cytoskeleton. It is the microtubule protein which is present in particularly the animal cell. The cilium, it is the movement in or of external medium, that is the critical development and signaling pathway, and its structure is that microtubule protein. It is mainly made up of protein structure and it is present in animal, protist and few plant material. Glyoxysome is another important organelle which converts the fat into sugar, and it is a single membrane compartment and mainly present is glyoxysome to the plant system. Melanosome is another organelle which is a single membrane compartment and it is a pigment storage organelle which is mainly present in the animal system. Microfibril is the bundle filaments which helps in muscular contraction and it is present in the animal cell. The vesicle which is also present in the eukaryotic cell and is a single membrane compartment and it helps in the material transport system and it is present in all eukaryotic cell. So, with these different organelles, I have tried to give you an overview of the different organelles and their function, how they perform different activities in the living cell, and I think you have now a little bit idea about this particular cell, eukaryotic as well as prokaryotic cell, and how they perform and how they work. So, with this today I am concluding my lecture. Thank you very much.